Hi, everybody. Welcome back. In the last video, we introduced the idea of protein design as an inverse protein folding or inverse protein structure prediction problem. Whereas protein structure prediction, protein folding starts with sequence as a given and asks the question, what is the native state? What is the preferred fold of that sequence? Protein design flips what is given and what is the target and asks the question, given a protein fold that we would like to achieve, what is a sequence or a set of sequence or the set of sequences that is able to fold into that state? We discussed how we can rephrase that question as an optimization problem, asking for what is the single sequence that best stabilizes the target structure, that is the single sequence that would have the best energy in the context of that target structure compared to all other sequences. And that optimization has interesting properties in that it's a mixed optimization, that there's part of it that's a search over discrete non-numeric variables of sequence. And there's another part that's a search over the real number dihedral angles or atomic positions corresponding to protein conformation. And there's this underlying constraint on all of this that we need to satisfy the protein structure. What we're going to be doing in this video and the couple subsequent videos is now digging into the algorithms that let us tackle this optimization problem. So let's begin by switching over to the board where we can start exploring this a little bit more. Let's start off by just sort of being clear on this optimization problem we're trying to solve. Recall that we were able to phrase the problem we're dealing with as this kind of apparently nested optimization, where we have a minimization over the set of all sequences, and what we want to minimize is the energy of that sequence in the target structure, but we can't define an energy of a sequence without some concept of the conformation. And so we can tackle this by adding in another minimization over the set of conformations of sequence S. And what are we then minimizing inside this inner loop? The energy of that conformation subject to or given the backbone constraints and any other constraints that are the target of the design. Now, there are two general strategies that we can take to tackle this problem. One is a nested optimization approach, and the other is what we might call an integrated optimization approach. In the nested optimization approach, we separate out conceptually the optimization over sequences and the optimization over conformation. And we can think about having some outer search over sequences. And this search over sequences is driven based on the energies that we get out of a search over conformations. And the search over conformations gives us a best conformation for the sequence, and we use that information. We use that information about the best conformation for that given sequence in order to inform the search over sequences. The integrated optimization approach doesn't make this distinction. Instead, takes a conceptual framework where we simultaneously search over sequences and conformations. And we don't really make, importantly, we do not need determine or evaluate the best energy for all, say, explored sequences, all sequences that we consider. Most of the protein design algorithms that are used, that we will be discussing, have generally fall within this integrated optimization technique. And this is because sequences and conformations are not independent. And in some cases, it may be possible to kind of figure out that a certain sequence just isn't going to go anywhere. A certain sequence isn't a good choice. And we may not need to actually figure out its best energy to be able to make that decision that it's a bad choice, right? Now, some methods have a little bit of a flavor of this nested optimization, that they're not completely integrated, that all sort of moves are that have happened, that there's a complete melding of the two ideas, and there's a little bit of a nested structure to them. Other methods really are truly integrated, that we really don't make a distinction between sequence and conformation within the algorithm, but we'll get to that as we move forward. I'll try to highlight with each algorithm which of these two classes it fits into, or how much of the degree of kind of the spectrum of methods from fully nested to fully integrated the methods sit into, or may sit into. All right. Now, once again, the big challenge in this problem, one of the things that we absolutely need to take into consideration and is going to affect the choices that we are able to make, is that these two optimizations are on different types of variables. The outer is a search over sequences. These are discrete, non-numeric entities, and the inner optimization fundamentally is a search over atomic positions or dihedral angles, is a continuous real numbers are the variables. And these are different kinds of entities. We need to factor this in the methods that we use. So the first approach that I would like to introduce is one that we've seen before. And we've seen it before in the context of conformational search. If you read any literature on optimization, and you will come across this. Almost any optimization problem can be solved by Monte Carlo methods, and in particular, to be specific, Markov chain Monte Carlo approaches. How do we use these Markov chain Monte Carlo approaches to solve a problem in protein design instead of protein structure prediction? Well, let's remember, just recap quickly what the basic MCMC approach involved. It involved an iterative approach where we would start with some initial guess, and then we would take a random move and compare the new state to the old state. In our approach, we used the metropolis criterion for that. What was the metropolis criterion? We looked at the ratio, so E to the negative delta E over KT, where delta E was E new minus E old. And we made this probabilistic acceptance or rejection based on this. And we essentially made a comparison between some random number generated on the interval from 0 to 1. And we compared it to, let me go back. We generate, just I want to give myself a little bit more space. We would generate a random number between 0 and 1, and we would compare it to the minimum of 1 or this number here. And this, we would ask, is this random number less than this metropolis criterion, right? And what this means is that if the change in energy was negative, that is, the new energy was below the old, then this would be a number greater than 1 and would always be greater than the random number on 0 to 1, so we would always accept. And if the energy was uphill but close to the old energy, then this would be a small positive number. E to the negative delta E over KT would be a number close to 1 but a little bit small, would usually accept. And if there's a big uphill move, the new energy is much larger than the old energy, this would be a number close to 0, and it would be very, very unlikely to accept the move. Right? Now, in the context of protein structure prediction, we did this with random moves that just involved changing a dihedral angle or changing a few dihedral angles or changing a bond length or a bond angle, changing something about the structure only. There's no reason that we have to limit ourselves to this kind of random move. And so in the context of protein design, we can really map this algorithm onto the protein design problem simply by changing what we mean by our random moves. Our random moves may include a change in sequence, that is, changing the identity of one amino acid or more than one amino acid at a particular position. Or it may include a change in conformation with the sequence unchanged. And there's no reason that we can't do this, right? There's no reason that we can't simply introduce the idea that the random moves may allow us to change the identity of an amino acid as well as the identity of as well as the conformation. This would allow us to walk both through conformational space and through sequence space in order to find our best combination of sequence and conformation. And you might ask, how do we implement constraints in this? Remember that it's important that we implement constraints. Remember that the constraints in the protein design framework are often that we have a backbone structure that we want. The constraints may simply be do not allow moves that affect the target structure. This is actually a very easy thing to implement. We say we start with a backbone that we want. And when we say that we allow moves that allow changes in conformation, we only allow moves to the side chains that are a part of the design. We don't include the backbone degrees of freedom as part of the allowed move set. And so this approach, very easy to implement constraints of portions that we can't move, easy to incorporate both sequence and conformation in the variation. And we can furthermore frame this general Monte Carlo approach within a simulated annealing framework. To change this rather from simply an exploration of the space into a true optimization. So if we were to do this problem to implement this Markov chain Monte Carlo at constant temperature, is not really going to end, right? It's going to be an exploration of this sequence conformation space, but without giving us a well-defined minimum. Now, it may be that we get kind of lucky here and find our find a minimum structure, or analyze our set of sequences and structures to find that minimum, but it's not obvious where it is there. However, Monte Carlo coupled with simulated annealing transforms this truly into an optimization framework, right? Gives a well-defined, best sequence and conformation, at least as long as we take the temperature to some limit close to zero, right? So we start at some initial temperature and we move to some final temperature that is close enough to zero that we actually have no more variation in our structure. And it often is the case that if you really want to frame this as a true optimization, you take it all the way to zero, just to be sure of what your answer is. Now, this is certainly easy to conceptually think about. It's a fairly easy thing to implement. There are some important caveats. First of all, when we're varying sequence as well as structure, unlike the case in molecular simulations when we're just trying to understand the conformation of a single structure, in that context, temperature means really the same thing as it does in everyday physical chemistry and in everyday language, that we expect that the distribution of states at 300 Kelvin to be roughly similar to what we actually see in a room temperature consideration, right? Or a body temperature consideration. In a, however, and at lower temperatures, we can interpret them based on our everyday experience. It's not at all clear that when we are varying sequence and the number of atoms are changing, and we have this additional variation, that temperature means the same thing. And so temperature may not have an intuitive meaning. That is, and this is generally true. We can apply these Markov chain Markov chain Monte Carlo methods to any sort of problem. We can apply the metropolis criterion for how we accept or reject moves in any kind of problem. But what temperature means, right? Temperature means in the context of this algorithm is a scaling factor on the energy, right? That's really what the effect of temperature is. It's this, it's this scaling factor of the energy. We divide the energy by KT. and it kind of sets the meaning of those energies. When we're dealing with molecules and the energies are in kilocalories per mole or another kind of natural chemical notation, then temperature might mean the same thing. But if we use this approach for something that then moves away from the physics, the physical chemistry in which we define temperature, then the meaning of temperature doesn't have the same relationship to our everyday experience. We can still use it. We can still use it as a scaling factor. We certainly can still use it in simulated annealing. When it's high, it makes uphill moves more easy to accept. When it's low, it pushes us only in the downhill direction. But what is high, what is low, no longer has that meaning. We think of it purely as a scaling factor for the energies. And we may need to do experiments, play around with how different temperatures affect the optimization protocol in order to get reasonable results. This exploration is something that is always important in the context of simulated annealing. How high is high enough? How long do we need to cool? These are very, very important and open questions. What is the appropriate cooling protocol? That is, what temperature, what's our initial temperature, how high do we start, how high do we end, how slowly do we cool in between? And again, in protein structure prediction, we are guided by the funnel-like energy landscape of protein folding dynamics. It is not at all obvious that that same funnel-like structure exists in sequence space or in the energetics around a target structure. And so it's not simply a case that we can transform and move a cooling strategy from the conformational search problem into the design problem. We have to address this separately for each piece. All right. So that's one caveat here. Any other notes? So we talked about this question of strategy, of a nested strategy versus an integrated strategy. Monte Carlo approach can really be applied in either of these methods, right? It can be applied in a sort of fully nested approach where we make a random change in sequence, then we fully explore the conformations and use the best energy to accept or reject that sequence. This would be a fully nested structure, all using Monte Carlo, but where we're making Monte Carlo moves in sequence, and then we're doing a full Monte Carlo exploration of the conformational space before we accept or reject the move. It can be truly integrated, where the moves are in sequence and conformation happen in a interleaved fashion. What do I mean by that? It means that really at any single sort of move point, you might choose a move in sequence, you might choose a move in conformation, and they might not be balanced. One might be more frequent than the other, but there's no real structure requiring one or the other. It's also possible that you might have something in between where perhaps there's a structure where you make moves in sequence intentionally less frequently, and you always make a certain number of moves in conformation before you make another move in sequence. And this would be kind of somewhere in between. This Monte Carlo based method of protein design is something that is absolutely used. Monte Carlo exploration of protein sequence is one of the, sorry, let me go back here, is one of the core algorithms underlying the, oops, sorry, thought I had my pen, is one of the key algorithms underlying the Rosetta protein design software package, which has successfully been used to design all sorts of different proteins, and is quite a powerful package. Okay, Monte Carlo is a kind of generic optimization algorithm. It works for essentially any optimization problem. There is no optimization problem that you can't treat in a Markov chain Monte Carlo approach. And so because of this, it's a go-to for many, many applications. It can be very efficient, especially when coupled with simulated annealing protocols and a metropolis criterion. But the fact that it is generally applicable and can be applied doesn't necessarily mean that it is the best choice for all problems, and there aren't other approaches. So the next algorithm I would like to introduce is kind of an interesting one, especially in the context of protein design, as it's an algorithm that we're applying to biology, but where the inspiration from this algorithm itself comes from biology. And let's think about other discrete optimization problems. Well, one pretty important discrete optimization problem in biology is that of evolution. Well, fundamentally, evolution is a kind of search for at least sort of locally optimally fit sequences. We can think of this as a search for kind of optimally fit sequences over time. But again, it's sequences, changing the genetic sequence of an organism, which is fundamentally a discrete system. You have your genetic code is made up of discrete individual nucleic acid choices. The set of options is vast, but it's a discrete non-numeric set of choices. Right? So we might sort of think of evolution is a discrete non-numeric optimization process. If it is, and if we think of it this way, perhaps we can learn from the algorithm of evolution to design an algorithm that we can use to solve problems like this. And this idea leads to the concept of what we call genetic algorithms. All right. Now, when many people who are introduced to genetic algorithms are introduced to them in the context of optimization problem alone, or in the context of computer science, in complete isolation from biology. We're a class looking at computational biology. Many people have a deep understanding of how evolution works and how genetics works. Genetic algorithms are not based on real evolution. They're not based on real genetics. They are inspired by genetics and genetic molecular genetic evolution. It doesn't really, for the context of the algorithm, it doesn't matter that we gloss over details, that we take an ultra simplified view. It gives us something that works. And so in this kind of very, very high optimization perspective of genetic evolution, let's think about how natural selection and evolution works. All right, so let's see, switch pen colors here for a minute. We'll get going. Okay, so let's take a look at this ultra simplified view of molecular evolution. So the first key point is that an organism is defined by a set of genes. And these genes are linearly arranged on a chromosome. So we might think of this visually. We have some long chromosome, and arrayed along this are individual genes, right? This is point 1. Describes the structure of how genetic information is stored. The second important point describes how variation can be introduced into organisms. Genes can mutate, right? An important aspect of genetic mutation, there are 2 important aspects of genetic mutation, is that mutation is a random process both in terms of what gene mutates and in terms of the change made to that gene. There's often pictures of evolution as having some, in popular culture, as having some directionality, an idea that evolution moves towards some form. That's not how evolution works. The individual steps are purely random, and there's and natural selection guides that based on some optimization framework, but there's an underlying random process. So genes mutate in a random way to give variants on that gene. Right? That gene. And this describes how variation can happen. There is another essential part, and this is that of, sorry, I want to do this in the same color. The third component is a question of fitness. Fitness is a concept defined, by all genes. That is, there's some integrated function of all of the particular variants of the genes that a particular organism has that indicates its fitness, and fitness impacts likelihood to reproduce. With more fit individuals more likely to reproduce, and less fit individuals less likely to reproduce. Right? And so you can imagine a process by which you have a group of organisms that have certain genes. These genes vary over time due to mutation, and if this random variations lead to increased fitness, that increases their chances of reproducing. The random variations that lead to decreased fitness lead to decreased likelihood of reproduction. But there is one other really, really important part of how evolution works in sort of natural systems. And this is the idea of sexual reproduction, which at the molecular level is represented by recombination. So let me. So our fourth essential piece is that variation isn't something that is isolated to an individual. We combine the effects of information from individuals at the genetic level through recombination. This combines genetic information from two parents. Now, how does recombination work? Recombination involves the idea that we start with 2 chromosomes. So we might have parent A. And parent B will have the same number of genes because it's a closely related organism, same species, but the identity of these genes will be different. Some of them might be the same. They may all be different. But we have 2 parents. And in the process of recombination, what happens is that the information of these 2 parents is mixed by making crossovers. And so there are points that the 2 genes, and physically this is actually what happens in the process of recombination, the 2 chromosomes line up next to each other. There are breaks made in the strands, and then they re-meld with the opposite strand. And the result of this recombination event is that we have progeny that are of mixed types. And so we end up with this recombination event that we start with 2 parents, and we end up with, we walk along to create the gene for 1. We walk across, we follow the crossover event, we walk along the second parent, then we walk along the second crossover event, and then back. And so this first of the progeny would have the first 2 genes with the variant from parent A, and the second and the middle sort of 4 genes with the variant from parent B, and the second progeny would walk along in the other way, beginning on parent B, following the crossover, walking along, following the crossover, and across. And this gives rise to progeny that have the middle 4 from parent A and the outer genes with the variant from parent A. And so what we end up with is 2 offspring or progeny, offspring 1, and just an offspring 2. And just as the process of genetic mutation where an individual gene is varied, the location of recombination events is also a random process. Which this means is that every time a pair of the same parents reproduce, they create new combinations of offspring, because the way that the parental genes are mixed are different. And this is why siblings from exactly the same parents aren't identical, right? Okay. So this framework involves a few, this sort of framework describes what happens at the individual level. An essential idea of evolution, and this is as much of an important aspect as any of the rest, is that evolution occurs, or I should say acts on populations. Natural selection acts on populations. Fitness isn't an absolute, it's something that's relative. In any sort of ecological system, there's competition. And why is there a concept of fitness and natural selection? Well, it's because of this competition, right? In the absence of competition, every organism would have perhaps an equal likelihood of reproducing, unless their defect in fitness was actually fundamentally a defect in the capability of reproducing. But many times, fitness is defined by things like a bigger organism might be more likely to get a nest because it can chase away the competition. And if it has a nest, then it can reproduce, right? Whereas the less fit organisms are less able to find a nest and thus can't have, are less likely to reproduce, something like that. So evolution acts on populations, and natural selection favors reproduction of the more fit at the current point in time. That is, it's relative to the population, right? It favors the more fit within the population. There is no absolute sense of fitness. Everything is relative, right? And over time, over evolutionary time scales, the population, so over evolutionary time, the average fitness of the population increases. Now, of course, in the biological context, this only happens if the actual definition of fitness is constant, which it isn't. Environments change, climate changes, all sorts of things happen. And so what it means to be most fit changes through time, and that affects the evolutionary dynamics. That's not something we need to be concerned with in the idea of a genetic algorithm. Now, why is evolution so effective, right? Why is evolution able to find such a diversity of solutions to different ecological problems? Well, part of it is the sheer immensity of ecological timescales, right? But even on shorter timescales, we see evolution happening on short timescales as well, right? It's effective not just because of the randomness, right? That is certainly part of it. There's a reason that sexual evolution, sexual reproduction evolved, why recombination is a common feature in reproduction, and that we kind of have two different modes of searching the space. We have random variations to individual genes that modulate function. You can think about these as small little changes. If one gene in an organism's genome has a change, it's a pretty small effect overall. Maybe it leads to improved function, maybe it doesn't, so on. But what does recombination do? Recombination takes two different solutions, if you will, two different individuals that are both equally fit, right? Or close to equally fit, and then takes their features and scrambles them and mixes and matches, and then creates new possibilities of two good solutions. And that recombination takes bigger jumps through space than individual random mutations do. Individual random mutations affect a single gene. Recombination affects all of the genes or many of the genes in an organism simultaneously. But whereas introducing a massive number of purely random changes everywhere in the organism is very highly likely to lead to lack of fitness, introducing recombinations, right, of equally fit combinations of equally fit organisms increases the chances that those larger jumps are still fit, and perhaps even more fit. It also allows variation to be maintained if there's parts of the structure that can't move. Maybe there's genes that the population has found the single optimum for, and any variation to that gene is going to lead to a decrease of fitness. Well, that will lead to a population where that gene is always present in every individual, and recombination will never mess with that, right? Because it only works with what's in the existing population, right? And so this using parts of proven solution gives a bias in the larger jumps through space, which may be particularly efficient. So how do we actually make an algorithm out of this? Well, we really just follow exactly the same steps. We can call things genes, and we can call things chromosomes. We can pretend we're doing mutation. We can pretend we're doing recombination. We can pretend that we're reproducing and applying the features of natural selection. But we do this with the parameters of our problem, right? The values of our variable. So what do we mean by this? Let's just walk through this for the protein design problem. Protein design with genetic algorithms. So the first thing that we need to do is we need to define what a gene is. Right? What do we mean by a gene in the context of a genetic algorithm for solving an arbitrary problem? A gene is one variable, or perhaps one variable, or a set of interdependent variables. Right? And the genotype is the value of these variables. So in the context of protein design, perhaps a good possibility may be the gene may be one position in the polypeptide, and it might be described as a amino acid type, and the side chain conformation of that amino acid. Right? We may want these lumped into one gene, one combined variable, because conformation and amino acid identity are not independent. They don't even all have the same number of degrees of freedom. A lysine has 4 rotatable bonds. An alanine has no rotatable bonds. So for alanine, there's no conformational variation. You've chosen an alanine, you know the conformation. For a lysine, you do have this concept of the different conformations. So there's a great diversity. Proline has a different set of conformational possibilities. It's a unique structure that bends around and touches the backbone. It has a different set of allowed variations than something like, say, serine. And so there's a coupling between amino acid type and conformation, and so it might make sense, and even at the even just the representation level, and so it might make sense to include those as one. So then what would, if once we have the idea of a gene, we can introduce the idea of mutation. Right? What would a mutation be? Well, a mutation is a change to the value of one gene, that is, these set of variables. In our context, there's 2 possibilities. One may be a change that takes us from one choice of amino acid and a conformation associated with that to a new amino acid and a conformation associated with this. And perhaps we may store some of the information about the conformation if it's possible to, or maybe we would generate a new random conformation, but a random change that includes the identity. Another type of variation that we could make would be a change that maintains the identity of the amino acid, but changes to some new conformation R prime that changes the conformation of an amino acid without changing the identity. With this, if each gene represents a combination of amino acid and its conformation, a mutation allows us to make changes to these. What would our chromosome be? Well, this is simply a list of genes arrayed linearly, which is the identity, I should say genes with genotypes. And this is precisely the identity plus conformation for the side chain of all positions. This is if we add in the knowledge of the backbone. Sorry. If we add our target backbone, gives us a full sequence and structure. Okay? We have an identity for every amino acid at every position. We have a conformation of every side chain at every position. If we add in the backbone that is known as a sort of a fixed part of the problem, that gives us a whole structure. So in this case, the complete chromosome is a single structure of a single potential protein. That is, we can think of this as it's a list of R1 with the conformation at R1 and R2, the conformation at R2, and so on to Rn and the conformation at Rn. It's worth noting that although this may be the ordering of these residues, may be the ordering in sequence, it need not be. What's important for a genetic algorithm is that once we have set an ordering, we keep that fixed. It may be that we want to choose an ordering that doesn't match on to the natural ordering. Maybe we want an ordering that keeps neighboring positions in the target fold closer together in linear sequence or not. And so there may be reasons we want to adjust the ordering. We might want to just try a random ordering. But once we've chosen order, it has to be fixed. So once we've done this, we can then start thinking about how we actually structure the algorithm. Right? How do we actually tackle this problem? First step is that we randomly populate some number of chromosomes, some n population, right? And I'm going to call this this set and I call this the population set. Right? And so this is really can be done purely random. You just throw in random sequences with associated conformations. Then what do we do? The next step is a reproduction step. We generate offspring. And how do we do this? Well, there's two things that go into the generation of offspring. We introduce random mutations. That is, in the process, each sort of parent has a possibility that it can have random variations introduced at each individual position, that change of amino acid, change of conformation, for example. We introduce random mutations into parents. And we apply recombination between parents. And we do this so that we have some trial set or some offspring set where we might call this set of offspring. Let's call this offspring set. And the number of these, the number of offspring, is much greater than the number of our original population. That is, we produce many offspring from a smaller number of parents. The next step that we're going to add in is a fitness calculation. We compute fitness. And in our case, since what we want to do is optimize the energy, we simply say that our fitness is the energy of our state subject to or constrained by the backbone. That is, we kind of compute the energy of our choice of amino acid sequence in the context of the backbone, right? Really just a molecular mechanics energy. We can think of this as the energy of our set of choices for a particular sequence and our given the backbone structure. And it's computed exactly the way we would compute a regular molecular mechanics energy. Now, then what we do is we select from the offspring set number of individual of reproducing individuals. Select N population individuals from the offspring set to reproduce. And this has to be done in a way that is biased by fitness. And once we've done this, then we can iterate. We now have a new reproduction set. This then becomes our new S population, and we then go back to the offspring generation set. And we do this over and over and over again, either for a fixed number of generations or until we have a final answer. Now, there's a lot, this is a conceptually a fairly easy approach. There's a lot of potential variations in here. There's a question of what is the frequency with which we should introduce random mutations. Similar to the question of how do we choose random moves in a Monte Carlo set? What's the frequency of recombination? What's the size of the original population? What's the size of the offspring population? In terms of how we sort of apply natural selection, the naive way is just to rank order by fitness and only take the absolute best reproducing individuals. That can also be done in a probabilistic way, where we randomly choose individuals to reproduce or not, but in a way that is biased in favor of improved fitness. These are all subtleties that certainly can have an impact on the performance of the algorithm, but the structure is always there. All right. Monte Carlo and genetic algorithms, actually, you might notice that they have some similarities between them. They're both fundamentally grounded in the idea that we randomly vary structure. They have a bias towards lower energy, but not necessarily a guarantee that we move towards lower energy, especially if the selection part of a genetic algorithm just sort of chooses in a probabilistic sense individuals to reproduce or not. There's a possibility that less fit organisms still can reproduce. It's just less likely. So there is part of a conceptual similarity, but there's also some big differences. Monte Carlo fundamentally tracks a single path through towards evolving better energy or evolving fitness, whereas genetic algorithms have this concept of a population. Because they have the concept of a population, genetic algorithms allow this recombination step, which isn't something that is part of the move set allowed in Monte Carlo. And these features of genetic algorithms make them very efficient in many applications, just as Monte Carlo is a very, very generic optimization approach that can be applied to almost any optimization problem. Genetic algorithms also are in that same framework. We can take any optimization problem and define each variable as a gene and think about random changes to those variables as mutation and recombination. And we can do this for any optimization problem. Both genetic algorithms and the Monte Carlo approaches through simulated annealing are very general, very powerful, and can be very effective. Both of them also have absolutely no guarantee that they actually give you the true global minimum answer. And both of them have a lot of parameters that you need, well, I shouldn't say a lot, but have parameters that you need to tune and can affect the quality of your answer and the amount of time it takes to get to them. In simulated annealing, the two key variables there, parameters that you can tune, are the move set, how do you choose the random moves, and the cooling strategy for the simulated annealing. In genetic algorithms, you also have kind of the move set question of how do you generate the mutations. You don't have this same sense of cooling, but you do have the parameters or the tunable parameters of the function or of the method, the mutation rate, the recombination rate. If you make those too slow, it takes a very, very long time to get an answer. If you make them too large, you can be introducing too much random variation, and you can't converge to an answer. And there's a happy medium in there that needs to be found by exploration, experimentation. Okay. So I'm going to end this video here after introducing these two possible general algorithms that we can use for any optimization problem, including protein design. In the next videos, we're going to introduce some more specialized algorithms that only work for certain structures of problems, but structures of problems that it turns out protein design maps onto quite nicely, and that I personally think are very elegant approaches to solving the problem. Until next time, take care and be well.